Thanks. So, yeah, as he mentioned, I'm a postdoc at the Mayo Clinic and Dr. Jim Maher's lab. And today I'm going to tell you a bit about some work we've done over the last couple of years with Aptamer-directed siRNA therapeutics delivery and a tumor called cholangiocarcinoma. And I won't bore you with too much clinical background. Okay. Just very briefly, a little bit about the tumor we're interested in. So cholangiocarcinoma is a tumor of the cholangiocytes in the bile duct. So they're the cells that line the bile duct here, and they reside either inside the liver. So we have intrahepatic bile ducts or extrahepatic biliary tree here. And you can develop tumors anywhere along the biliary tree. And the tumors are also broken down anatomically among these either extrahepatic or intrahepatic tumors. And these classifications matter because it really affects the outcomes for patients. So among patients with these intrahepatic tumors that are localized and totally within the liver up here in the upper part of the biliary tree, the outcomes are okay as far as this cancer goes. If you're eligible for a full liver transplant, you have roughly a 50 or 60% 5-year survival rate. But for other cases, the survival rate is very, very poor. And in many cases, the tumor is detected way too late to operate on, as is often the case with tumors like this. So most patients have no curative treatments available to them. Gemcitabine and cisplatin is the normal chemotherapeutic route that people will take, but that usually gives something like a 9-month life extending benefit, and it's never ever curative. So what we're interested in is trying to develop new systemic therapies that could be curative or could maybe improve the effectiveness of the chemotherapeutics that are already given to people with these tumors. So some of our collaborators at Mayo, Rory Smoot and Tushar Patel, are interested in the cancer biology of these tumors. Rory is a surgeon who works on cholangiocarcinoma, and his lab maintains a lot of PDX models of these tumors. And Tushar is a transplant hepatologist, so he specializes in transplant medicine for people who have had their livers transplanted after having a tumor. And something that they discovered in several models of these tumors is that they're susceptible to siRNA knockdown against a very highly evolutionary conserved pathway called the Hippo pathway. So it's called this because it was actually found in Drosophila originally, and usually we name things in Drosophila after their phenotype. So the genes in this pathway are called Hippo because if you dysregulate this pathway, the flies get huge. And this is the same thing that happens in tumors. If you dysregulate the Hippo pathway in cholangiocytes, the cholangiocytes get larger, they replicate really quickly. And this is typically associated with an event in FGFR signaling that upregulates this pathway. Very, very common in cholangiocarcinoma and some other hepatobiliary tumors. So they developed this cocktail of 3 siRNAs against 3 different steps in the Hippo pathway. 1 is against a kinase and 2 are against transcription factors. And here, when they treat these SB1 model cells with in a colony formation assay here, we have this control, of course, where we have colonies formed. The standard of care chemotherapy, gem-cis combination reduces cell growth of the transfected siRNAs in combination do the same. But the best effect comes from if you the delivery of these 2 together. But the obvious question that we now have is how do we actually get these siRNAs into tumors inside of people, and how do we get them into the right place? Because we don't want to dysregulate the Hippo pathway all over the body. So our strategy for doing this is to work with these 2 groups to deliver these siRNAs with Aptamer-directed therapy. So our group develops the Aptamer and aided in the characterization of this. Tushar's group provides these lipid vesicles that we use. They're simply transfected with the siRNAs, and then we can use during synthesis, we can have a cholesterol put onto the 3' end of any aptamers we select to target the tumor, and hopefully deliver these to mouse or PDX models of tumors and see if we can knock down the gene targets in these tumors. So the selection we performed did 10 rounds of selection using totally unmodified 40 nucleotide random region DNAs. And we did a toggle negative selection in this case because, as a lot of people are familiar with, liver cells soak up everything. So in even rounds, we did negative selection against this liver cell model, HU1545. And in the other selection rounds, we did negative selection against normal cholangiocytes. And in all cases, the positive selection here for all 10 rounds was done against this human model of cholangiocarcinoma, HUCCT1. So we found 9 candidates that we wanted to screen. I'll just show you one result of all the characterization we did on this pool to select an aptamer. In this assay, we synthesize these aptamers with a 3' biotin, stain them on cells for a bit, and then we use an IncuSight instrument to take multiple images over multiple wells in an unbiased way. And we can stain the aptamer with a fluorescent streptavidin and just measure the binding to the target cell line. So these are the cholangiocarcinoma model cells here. And we have a few aptamers that are statistically significantly enriched versus a scrambled negative control if we just measure binding this way. We took some other approaches to confirm some of this, but this is the first pass of what we did with the candidates we selected. Based on some other data that I won't show here for the sake of time, we also saw that the aptamer number 1 at the top had some selectivity versus liver cells in vitro. So we picked that one to use moving forward. I also have here just quickly some microscopy showing that these aptamers, so we have a fluorescent label on the aptamer here. These are again HUCCT1 cells where we just have actin labeled in red. And these seem to, for the most part, stain the membrane of these cells. These aren't internalized. There are a couple of cells in here, like this one maybe, where there's a small amount of internalization, but it seems like whatever this aptamer is sticking to is on the surface of these cells and is probably not being trafficked in much, if at all. So the first question we asked was, of course, what happens if we inject these into animals? Do they actually home to tumors? So here we have an orthotopic model of the tumor. When we say orthotopic, you can't really inject into the biliary tree of a mouse, but these are cells just injected directly into the liver. It's kind of the closest we can practically get. And then we label the aptamer with 5' fluorescein or a negative control, which is a scrambled version, and then inject this into the tail vein of animals, allow it to circulate for 4 hours, sacrifice the animal, perfuse, and then section tissues to see where fluorescent label ends up. So there is some fluorescence for both the aptamer and the negative control in the liver and the tumor, but the aptamer is very, very well enriched in the tumor versus the liver, which we were happy to see, and agreed with our in vitro data that I mentioned that shows that this aptamer did not stain the liver cell model in vitro as well. If we zoom out just a little bit on these tumors as well, you can see how well this actually stains the entire tumor. So the previous images were zoomed in on a single spot, but if you look at the negative control here, you can kind of see where the Dept staining has these compacted nuclei, and you can identify the border of the tumor. And while we don't see negative control scrambled aptamer in the tumor, it's localized the tumor here. And we're not sure what the target of these aptamers is, but it seems like maybe there is some of these blood vessels as well, because you can see a little bit of collection of the blood vessels for the aptamer that's not present in the negative control. We also wanted to detect aptamer across the whole animal by just breaking down resected tissues and measuring these by qPCR. So all our aptamers, including the negative control, keep the constant primer region on the outside, and we just shuffle the middle or use a selected aptamer. And when we resect a few tissues, we see if we just look at the orthotopic tumor versus the liver, this reflects what we see in the imaging, where it's very well enriched in the tumor. Unsurprisingly, there's some accumulation in the spleen. This is almost always the case with oligonucleotides. And then we have a little bit of off-target accumulation in lung and heart and a bit in the kidney, which is also not too surprising, but these are fairly well enriched in the tumor versus elsewhere. Another important question, of course, is going to be, do these actually localize to the right place if we include aptamer? If we include the vesicle. So here's the exact same experiment as I showed before with tail vein injection and circulation for 4 hours. But in this case, the oligonucleotides labeled in red, and in green, we have a membrane staining fluorescent dye that is in the membrane of these vesicles carrying the siRNA. And again, the vesicles and the oligonucleotide here, or our aptamer, localize quite well to tumors versus the negative control. And I haven't shown the data here, but if we compare this to, or if we look at qPCR for this data, it compares very similarly to the other qPCR I showed for tissues. We can also show target gene knockdown with these siRNAs. So we dose animals every 2 days with a single injection through the tail vein for 2 weeks. So they get a total of 7 injections of aptamer. plus vesicle plus siRNA. And we treated these mice with different combinations of either aptamer or negative control on the outside and siRNA or non-functional negative control siRNA loaded into the vesicle. And the important result here is in the last column where we see that only if we have functional aptamer targeting the vesicles, not the negative control, and only if we have functional siRNA against this transcription factor in the hippo pathway, but not the negative controls, we see significant knockdown in the tumors versus these other conditions. Although in all of the data I'll show here is pretty inconsistent from animal to animal. So there's an average effect, but some animals don't respond as well. If we compare this transcription factor knockdown in the adjacent healthy liver, we don't see a statistically significant knockdown. It is down numerically a bit, although it's not significant in liver. And then this is another hippo target pathway we looked at. So WWTR1 is a co-activator along with YAP in the hippo pathway. It is, again, knocks down in the tumor only when we have the aptamer and the siRNAs present, but not in the healthy liver. And then finally, LCK, this is a kinase that's related to activation of the hippo pathway. And again, we're able to knock this kinase down in the tumor, but not in the liver. And we're excited about these targets in particular because these are considered classically undruggable targets because you can't, there are not good small molecule drugs against these small transcription factors or co-activators. And some kinases like LCK can be really challenging to drug for systemic toxicity. So you can't just deliver a small molecule against many of these. Even if it will affect the tumor, it's going to be so systemically toxic that you can't actually use it as a therapy. So this was all well and good, but despite the fact that we knocked all these genes down in the tumor, we didn't see any reduction in tumor weight over a couple of days, over a couple of weeks. So there are a lot of explanations for why this could be. Of course, maybe we're not dosing enough or not dosing often enough. It's also possible that we need to maybe spend some time modifying these siRNAs for stability or to identify better targets. We're probably getting a cyclical effect every time we dose these. There's a knockdown, and then over a couple of days before the mice get the next dose, the gene expression is going to go back up, and then we knock it back down, and this might just not be effective over the course of 2 weeks. We may also need to look at other siRNA targets. These are kind of arbitrarily chosen because they're canonical and they're known to be important in the hippo pathway, but they might not necessarily be the best therapeutic targets in that pathway, even if the whole pathway is amplified. And then finally, something we're interested in the future as well is combining this with the GemCis treatment in the animals, which is kind of the obvious next step. So we were wondering, would the siRNA alone be successful in the animals to knock down tumor burden? And that's unfortunately not the case, but it might work with GemCis. So in addition, a couple of things we're thinking about additionally to what I mentioned. Something I'm really interested in is radionuclide conjugated theranostic applications. So the diagnostics for cholangiocarcinoma that are currently available are very, very, very poor. There are biliary tree scrapes, for example, that you can do, and they're about 50% sensitive, so they're not very helpful. Sometimes the clinicians will even do these endoscopies and scrape cells and look at a slide and see no tumor, and then you scan someone and you see a tumor. So the diagnostics are really not very helpful. We think that maybe these radionuclide conjugated versions could also be good for identifying metastatic sites, because you don't want to give a liver transplant to somebody with a bunch of metastatic disease that you haven't detected, because they're of course going to recur, and you're kind of wasting a liver that could go to someone else. And finally, the tumors in cholangiocarcinoma are really both molecularly and anatomically diverse. So we have several of these aptamers that we selected, and a few show in vitro activity, but I want to explore some of these in vivo with different tumor models to see maybe we need different combinations of aptamers for certain tumors, or maybe even on a tumor specific basis, there's a best aptamer for each model that we have. So with that, I just want to thank the lab and especially Caroline, who you're going to hear from next, who helped with a lot of the imaging that I showed, and then the SMOO lab as well was really instrumental to this, and Ennis and the SMOO group helped with the animal work.